Okay, I think we are ready to begin. Good morning, everyone, or good afternoon, perhaps. Welcome to the NCI Symposium on Mutation Signatures in Cancer. So over the next 2 days, this symposium will cover the state of the science of mutation signatures in several important areas: normal and cancer tissues, exogenous and endogenous processes, analytical approaches, and translational aspects. The symposium is chaired by Dr. Ludmil Alexandrov at the University of California, San Diego, and Dr. Terry Landy at the NCI. Can you advance the slide? Okay, and next slide. I just want to begin with some acknowledgements of people who were critical in planning this event. First of all, the planning committee shown here, representatives of NCI program staff and intramural staff from several different divisions. Financial support was provided through the Division of Cancer Biology, of which Dan Gallahan is the director. Claire McCarthy was critical in many ways in helping bring this meeting together. So a special shout out to her for the communications aspect. And then finally, the conference services, Mike Cavanis and Terrence Calhoun at Rosalie and Associates have put together the meeting website and conference organization. So before we begin, there's several details. So for the talks, attendees can enter questions for the speakers in the Q&A box, which is at the bottom of your screen. And these can be addressed during the 5-minute question and answer period that follows each talk. There is closed captions that can be activated by clicking the CC button at the bottom of your screen. If you have technical difficulties or questions, please contact Mike Cavanis, who you can go to the meeting registration, the meeting site, and then on the technical assistance page to contact him. Do plan to attend the poster session later today. It's between 1:30 and 2:30 Eastern Standard Time. Later today, a Zoom link will be emailed to all registrants for the poster session, and you can click on that to attend it. And there you can meet the poster presenters in breakout rooms using the Zoom platform. In addition, the posters can be viewed at any time from the meeting website. And so now I'll turn over the program to our symposium chairs, Drs. Alexandrov and Landy.
Thank you so much. Thank you so much, Ron. And on behalf of the chairs, I want to welcome you. I'm going to start by giving a brief introduction to mutational signatures. So everybody has some background, and then I'm going to turn it to my co-chair. And when we speak about mutational signatures, we usually need to speak about somatic mutations. And as most of us know, somatic mutations occur in every single cell in our body, and they accumulate throughout our lifetime. And somatic mutations may accumulate just because the cell is dividing, or just because the cell is operating normally, for example, by generating energy, which causes reactive oxygen species, which damage DNA and mutate it. Somatic mutations may accumulate due to lifestyle, environmental exposure, such as exposure to ultraviolet light, due to lifestyle choices, such as smoking tobacco, cigarettes, and they can also accumulate through unknown and known carcinogens and mutagens. And here what you see is the molecule of aflatoxin, which may be consumed in knowingly or unknowingly. And again, that will mutate certain cells in the body and will increase the risk for developing cancer. And obviously, we have all the things that can attack the genetic integrity of cells, but through time, through evolution, our cells have developed DNA repair pathways that protect that integrity. So we have that constant battle between mutations accumulating or DNA damages accumulating and the damages being repaired, and that battle is slowly lost and mutations accumulate in different cells. But when we talk about mutational signatures, we first need to talk about mutational patterns. And the mutational patterns is what we observe when we sequence a cancer cell or a normal somatic cell or a precancer. And we have known that there are specific patterns of mutations in human cancer for a very long time. In my mind, the landmark studies that have shown that were the ones from Monica Holstein, Bert Vogelstein, Kurt Harris, specifically focused on TP53 in human cancers. And this is a paper from 1991, and showing that different environmental exposures generate different patterns in TP53 or in exons of TP53. And again, this paper was followed up a few years later, and it's a paper from 1994, again by Holstein and Harris, where they looked around the world in different cancer types, and they showed that there is different patterns of mutations, and they were able to link them to different environmental exposures. And not only did they show that there's different patterns of mutations, but they also show that there is different sequence context, transcriptional strand bias, or interaction between the mutagen and transcription couple repair, as well as a number of other intricate biological or DNA repair findings. But this was focused on TP53. And I just want to show you a summary of just two poster child cancer types. So if we look at UV light associated skin cancers, predominantly in TP53, we are going to see C2T mutations. This will be a dipyrimidines with a very specific strand bias. And again, if we look in tobacco-associated lung cancers, the majority of mutations will be C2A, again, with a very specific transcriptional strand bias. Again, this is mutations in TP53, and with the advent of next generation sequencing and the ability to generate whole genomes and whole exomes, we were actually able to start looking at a much larger genomic landscape. And the next set of, in my mind, landscape papers that show that was the ones from Peter Campbell and Mike Stratton, and Peter is our keynote speaker today. And this was a paper in Nature in 2009 that they actually showed the tobacco pattern in whole genome sequence lung cancer, and a back-to-back with that was a paper again in Nature, where they showed the pattern of mutations from UV light in a skin cancer. And again, that showed that these patterns are not confined to driver genes, they're across the genome. They were able to show a number of other exciting findings related to DNA repair, to transcription couple nucleotide excision repair. But essentially, this opened the door to examine patterns of mutations in genomes and subsequently exomes. But all of those are patterns. These are the things that we observe in the sequence data. So from patterns, we needed to move to signatures, and these are the things that generate the patterns. And again, I want to conceptualize that for you in regards to cancer, but one can do that in regards to normal tissue. So if you have a chemotherapy-resistant recurrence, and in that chemotherapy-resistant recurrence, one can have many mutations coming from many different sources. One can track the lineage back of this chemotherapy-resistant recurrence to the fertilized egg. And here in this schematic representation, where the cell is blue, it's during its normal lineage. Where the cell is brown, it's become after clonal expansion and after neoplastic expansion. And as you can see, we get many mutations accumulating from many different sources, and they can come from different processes. So one can have a process A, which has a specific pattern of mutations or specific signature. And in this case, I'm using colors to illustrate the point. So in this case here, mutational process A is predominantly these black mutations. And this could be something very weak, something that has been active throughout the lineage of the cancer cell. And we can have a second process B. We get another signature predominantly generating red and purple mutations. This could be something much more potent, something that started in, let's say, adolescence and stop when the cancer was discovered, or something like tobacco smoking will be a good example. And we can have a third process C generating, in this case, orange and green mutations. And this could be something stronger than process A mutagenically, but weaker than process B. And this could be something that started with neoplastic expansion. So something like ApoBec activation or microsatellite instability would be a good example. So in this case, we have three distinct processes. Each one of them has three different mutational signatures, and they're accumulating throughout the lineage of the cancer cell. However, when we sequence that chemotherapy-resistant recurrence, we're going to get only a single sample if we sequence once. And that sample is going to be a mixture of these mutational signatures, a jumble of the way they have been accumulated throughout time. And now we have the algorithms, and you'll hear more about throughout the day, of deconvoluting that and separating them. So from this mutational pattern of the final cancer genome, one wants to find the mutational signatures of the processes that generated that pattern. And indeed, that was the way to move from a mutational pattern to a mutational signature. If you have only a single mutational pattern and no other additional information, this is not possible. But because we had hundreds, thousands, and now we have tens and hundreds of thousands of patterns of cancer genomes, each of them are slightly different processes, this becomes a solvable problem. This becomes a blind source separation problem, which can be solved with something like non-negative matrix factorization. The signatures can be separated and quantified. And I can illustrate that with yet another example here, another illustration. We have a large number of cancer genomes. Each one of them, we observe different patterns of mutations. And by factorizing that or by applying machine learning approaches, we can find the different mutational signatures active in these cancer genomes. We can quantify each signature. We can say that many mutations are found in that sample. And we can never do that perfectly. There is always going to be some residual error. We want to minimize it without overfitting the data. I should say this is this problem we were able to solve in 2013 and publish the method at the time at Cell Reports. And then using that, we were able to generate the first comprehensive set of mutational signatures in human cancer, which was also in 2013. And this was further updated in 2020 with the current reference cosmic set of mutational signatures in cancer. And we continue to update that. Mutational signatures come in many forms. Probably the ones that are more familiar are single-base substitutions. I just wanted to give you a few, and people will talk about those throughout the talks, but there is at least 50 different mutational signatures that are associated with non-biological processes. We have things that are endogenous, such as things related to the activity of ApoB3 deaminases, and you'll hear more about that from Ruben Harris later today. We have signatures related to chemotherapy, and there's some wonderful work by Nuria's group describing different types of mutational signatures due to chemotherapy. And again, there is signatures of different environmental exposures, such as restolochic acid, aflatoxin, and again, we'll hear more about that in tomorrow's session, for example, from Steve Rosen. I want to say that the usability of mutational signatures, again, going from the pattern to the signature, and I just wanted to illustrate that with a single example. This is the example of a lung cancer patient, a tobacco smoker, and this is the pattern of mutations you see on the left. And on the right, you can see that that pattern of mutations can be explained using at least 5 signatures, and we can actually say how many mutations each one of these signatures generates. So for example, signature 1 generates 1100 mutations, signature 2, 7,000, signature 4, 19,000, signature 5, 6,000, signature 17, 2,000. And we now know the etiology of many of these signatures, and we can say the majority of mutations in these patients are due to signature 4, which is associated with tobacco smoking, and indeed, this is a tobacco smoking lung cancer. We can even take a step more. We can go to the driver mutations in the cancer and across many cancer types, and this is an example of KRAS driver mutations across a large set of lung adenocarcinomas. And we can say that big chunk of these KRAS mutations are actually generated by the tobacco smoking signature, for some by signature 5 and a small proportion by the ApoB signatures 2 and 13. So we get that extra level of resolution of what actually generated the driver mutations. The last thing I want to finish is in a way to summarize in this quadrant the capabilities and applications of mutational signatures. We'll hear a lot about AI development, machine learning development, different approaches. People have taken it to the next level to be able to separate DNA repair and mutation and DNA damage and repair processes from these mutational signatures, and we'll hear that during the machine learning session. There is a lot of work of using mutational signatures as for a better resolution lens into mutagenesis in normal tissues, in cancers, in model systems, and again, we're going to hear more about that throughout the days. Mutational signatures have become widely used for genomic epidemiologies. We have representative of the Mutograph Consortium and the Sherlock Consortium, and they operate in more than 40 countries taken together, trying to understand how cancer risk differs between different countries around the world using mutational signatures. And last and not least, the clinical applications. We'll hear about how mutational signatures can be used for clinical applications using things like Sigma and machine learning approaches for signature tree, which is the deficiency homologous recombination, but also hearing how our colleagues in the childhood cancers have utilized mutational signatures in clinical decision making. Lastly, I want to end, if you want to read more information, it's a very brief overview of mutational signatures, but if you want to read more, there's a wonderful review that just came out from Serena Nixano and her group in Nature Cancer. It's a very comprehensive and tells you a lot about mutational signatures, so I strongly recommend reading it. With that, I want to thank you for listening. And again, if you want to look at the reference set of mutational signatures, they're available at our Cosmic Mutational Signature website. And if you want to tweet, this is the tag for this meeting. So with that, I'm going to stop sharing and I'm going to actually switch to my co-chair, to Maria Teresa Landi, who is a senior advisor for genomic epidemiology and senior investigator at NCI, and she'll tell you more about the meeting. Terry.
Thank you, Ludmil. This was a really an excellent presentation. It's an overview of the historical use of the mutational signatures and also, the most current work. And I, again, welcome everybody. And this is the second event that the NCI has organized to discuss the mutational signatures in cancer research. The first was in 2018 and only involved a few investigators. And so I'm very delighted to hear that there are many participants at this time at this symposium. I heard that last week there were more than 850 registrations from 44 different countries. This is really a testimony of the interest that this approach has in cancer research. But what happens is that the more we delve into this type of topics in research, more questions emerge. And so although there was a lot of progress in the last few years, from the last time we met, in terms of developing new algorithms, the use of mutational signatures across different settings, issues that have been addressed in computational ways. But still, there are many questions, and we hope that we will be able to address at least some of these questions during this symposium and raise the attention about some of the remaining issues so that the scientific community can address these remaining issues in the near future. So in order to define some of these questions, let me show, I categorized the major questions in 3 groups. There are probably many more questions than this, but at least these are something that we see as important to start to address. First of all, the mutational signatures is being used through different settings. I listed here 2 major applications that are already mentioned by Ludmil before. So the etiological studies in epidemiology and the clinical settings. In terms of the etiological factors, etiological studies, the mutational signatures have revealed a series of risk factors, but there are still many unknown signatures that have been validated across different studies in the same cancer types, but we still don't know what are the risk factors associated with these signatures. And so this is a really important issue that we all as a research community should address and trying to reduce the number of unknown signatures through using different ways. We also needed to try to understand what is the impact of the specific features of the exposures and types of exposures in generating the signatures in different organs. So for example, what is the impact of an exposure that has very small amount of dose, but for a long time versus a very intense exposures long time before the time of diagnosis when the signatures are studied in the cancer. Or what is the effect of the age at exposure? So can an exposure that happens during childhood has a different effect in terms of generating signatures in comparison to an exposure during adulthood or so on? And in terms of the types of exposure also, are we talking about a specific mutagenic exposures, or some carcinogenic effects that is not directly mutagenic? Or do we need an impact of the carcinogen in the directly in the organ or through a metabolite? And what is the effect of mixture of signatures that can be very different across different geographical regions and so on? Also, we know that there are being already findings of different germline variants that are associated with specific signatures, like the ApoBec or the DNA repair damage signatures. But we know that there are many, potentially there could be many more. And so what is a way to systematically address the way in which the germline variations affect the exposures and the way in which the exposures generates the signature? And so for epidemiological studies, these are very important questions that need to be addressed. In terms of the clinical settings, although everybody now in the field, suggests that we could use or hope to be able to use the signatures to provide cancer patient stratifications for suggestion for therapy or prognostic features, we still need to reach a consensus on when and in which conditions we could use the signatures for clinical purposes. And we know also that there are treatment-related signatures. We will hear from Nuria, but how can these be used for prognostic purposes? And then very, very important questions is the potential use of FFP samples, fixed tissue blocks from biopsies that are typically used for targeted sequencing. Can we use these samples for identifying signatures, taking into consideration the fact that even the fixation of itself generates signatures? And is there a role of distinguishing the signatures between clonal and subclonal in terms of predicting the prognostic features and the risk of metastasis? And which are the orthogonal approaches that we need to use to validate the signatures, particularly if they have to be used in the clinical setting? And in terms of the validations, there could be different ways through laboratory or computational approaches. We know that there are different model systems for testing the exposures, but what are the differences by using specific cell lines or stem cells, organoids versus animal models, and how they can translate this into the human exposures? And are there integrated ways to look at different genomic features to better validate a specific signature? And in terms of the statistical or computational approaches, are there specific conditions or research questions that requires the use of supervised type of approach, particularly when we expected to find a specific signatures and we're looking for that specific signature, instead of using the typically used non-negative matrix factorizations or all the other methods that have been typically used in unsupervised fashion. And also, is there a way that computationally we can address these questions of the unknown exposures or the exposures that are determined in cases in which there are very low tumor mutational burden, or we have the featureless or flat type of signatures that are very difficult to distinguish from each other? And so what is the role, particularly in cases like this, of using deep learning approaches? And finally, best practices. We estimated there are at least 32 different algorithms that are being developed to date to identify different signatures. So we really need a sort of benchmarking, testing the different signatures, trying to define standardized way to use these signatures in specific conditions. And particularly also, we needed to define what are the minimum criteria that are required to identify the signatures in a robust way. So for example, is there a minimum number of mutations that we need to identify a signature, or a minimum number of samples? Is there a role for tumor purity in identifying signatures, particularly when we are talking about the flat signatures? And is there a role on looking at signatures at different organs because there may be tissue specificity and so on. And when we report these results in the literature, what are the minimum type of information that needs to be published so that the research community can replicate this work? And then finally, is there a way to create a repository where all the signatures, all the work that has been done and is continually updated? So any researcher can go in a specific place, test the different signatures and potentially answer each question. In my talk later on, I will spend just a couple of minutes to highlight some initiative that we're doing at NCI precisely to address this point. And then I move on now to define the program. So as Ron mentioned at the beginning, we have a very rich series of sessions, and we hope to be able to address these questions, or at least some of these questions, in the normal and cancer tissue, the analytical approach, exogenous and endogenous processes, and the translation aspects. And then at the end of these 2 days of work, Ludmila and I will try to wrap up what comes from the different discussions, also involving any of the speakers or the moderators, trying to define also the future direction for the research community. And now I have the honor to introduce the keynote speaker. Dr. Peter Campbell is the head of cancer, aging, and somatic mutation and senior group leader at the Wellcome Trust Sanger Institute in the UK. And he will talk about somatic mutations in aging and disease. Thank you, Peter, and thank you all.
Thank you very much for the kind introduction, Maria Teresa, and thank you for the opportunity to speak. I'm just going to share my screen. So I'll share that, and then so we should hopefully. Excellent. So hopefully you can see that. I thought I would present to you some of the work that we've been doing recently, focused on somatic mutations in normal tissues. And for today, I'll focus on liver, although there'll be some implications for other tissues as well. By way of background, Ludmila's already shown this slide, so no real need for me to introduce it. But I think, you know, we're really interested in these patterns of somatic mutations and the mutational signatures. Indeed, the whole purpose of this meeting is to kind of to collectively think about these issues. And you know, we've come at this historically from this kind of point of sequencing the tumor genome, and of course, you know, that tumor genome has accumulated mutations from all of these different mutational processes that are active throughout life, and many of those mutational processes are specific to the cancer. Many come from environmental and lifestyle exposures, and probably the majority come from intrinsic mutational processes. And what is interesting, what is difficult to do in the setting of sequencing a cancer genome is to kind of tease apart the temporal kind of aspects of these signatures when they're active. And one route that we've been exploring to look at this is to begin to look at tissues while they're still normal. And the advantage of that is that you can look at many clones from one individual, so you can spot whether there are clone to clone, whether there's clone to clone variation in the activity of these mutational signatures. And you can kind of look at how these mutational signatures play out across the lifespan, and how they're related to known exposures in those individuals. Now, sequencing normal tissues is a relatively challenging process. This is what 1 square centimeter of skin looks like in cartoon form, where each of these circles represents a clone or a subclone. And what you can see is that in normal aged tissue, the skin is particularly polyclonal. And you can imagine that if you took the square centimeter of skin, ground it up into DNA and sequenced it, you'd be sequencing DNA from all of these different clones, all of which would have independent sets of mutations, and therefore you would struggle to call them mutations. So the approach that we've spent most of our time on, and certainly will underpin the work that I present today, is to use laser capture microdissection and to sequence much smaller cuts of tissue. So what we're aiming to do is really to sequence down as few as 100-200 cells at a time, and to do that without any pre-amplification of the DNA. So we don't use any whole genome amplification. And that gives us very good quality genomes. It's taken quite a lot of effort to reconfigure both our wet lab protocols to be able to develop these sequencing libraries without pre-amplification, but also our dry lab algorithms to call the variants from these whole genomes. But that's the basic approach that we take. So I'm going to take you through some of the data that's emerged, particularly concentrating on liver and what the sort of implications of that are for our understanding of how mutational processes act in normal tissues. So the first point to make is that mutations start from the minute we're conceived. And so this is a study that we published earlier this year, where we took an 8-week fetus and an 18-week fetus, isolated bone marrow and hematopoietic stem cells from liver and bone marrow, then grew up colonies and performed whole genome sequencing, called the mutations, and then reconstructed phylogenetic trees. And then we could look at all of the other tissues from that fetus and essentially establish whether the mutations that we identified in these blood colonies were seen in these other tissues, and then use that to reconstruct the kind of evolution, the lineage tree of that fetus. And this is essentially the tree that we get from the 8-week fetus, and it's a fairly similar sort of pattern for the 18-week fetus, where we essentially build a tree, and you can see that this tree is typical of a very polyclonal tissue, like a fetus you would expect to be, where we have lots of independent branches. So each of these branches kind of finishes in one of the blood colonies that we sequenced. And this is the sort of lineage relationship. So you can see that, that there's a split right at the top of the tree, which represents the kind of first division of the lineages. And we have then colored the branches by the mutation. So each of these kind of branches contain some mutations, and we've then looked for those mutations in the other tissues from that fetus, and essentially we've colored them by which other tissues we see these mutations in. So these red mutations, the mutations colored in red, which you can see in this branch, and these branches here, are all seen not only in the fetus proper, but they're also seen in the trophoblast, in the placenta. And so these mutations must have pre-existed the separation of the trophoblast from the zygote, from the inner cell mass, as it were, of the fetus proper. And that tells you that these mutations occurred, essentially in the first kind of 4 or so cell divisions of life in this particular fetus. And what that means is that the mutation rate in the first cell divisions of the embryo are probably in the order of about an average of one or so mutations per cell division, inherited by each daughter cell of those initial zygotic divisions. So mutations really genuinely start from the moment we're conceived. And then unfortunately, as we continue through life, mutations continue to accumulate in every single cell, and they pretty much occur at a constant rate throughout life. So if we look at blood, lung, endometrium, even if we look at post-mitotic tissues, neurons, smooth muscle cells, and so on, we find that that the number of mutations steadily increases at a constant rate throughout life. The rate varies across different organs, as you can see here, but essentially every cell is accumulating mutations at a constant rate throughout life. So I promised to kind of concentrate on the liver, and the reason that liver is interesting to us is that it has a very well recognized pre-malignant phase that's associated with chronic liver disease. So liver cancer almost always emerges in the context of pre-existing chronic liver disease. And there are a number of different causes of chronic liver disease that all increase the risk of cancer, albeit to different rates. And those causes, relate in various ways to the kind of patterns of mutations that we see in the hepatocellular carcinoma that emerges. And indeed, when one looks at mutational signatures, hepatocellular carcinomas have a very rich landscape of mutational signatures, probably the tissue type that has the most variety of mutational processes active during its pathogenesis. And so it then becomes interesting to ask the question, well, if those are active in the hepatocellular carcinoma, when do those mutational processes emerge during this progression from normal liver through chronic liver disease to hepatocellular carcinoma? And that was one of the sort of fundamental questions that we were interested in when we started this. So we did our approach of taking laser capture microdissection from liver biopsies, and essentially we now have about 1,500 genomes from about 30 odd patients. This is a subset of the data here. And we've measured the mutation burden and the mutation signatures on all of these biopsies. And this is the sort of pattern that we see. So within each of these categories, the patients are ranked by age. Within each patient, we've done multiple microdissections, so we have multiple independent reads of the mutation burden from each patient. And so what you can see here is that when we look at overall mutation burden here, in this case base substitutions, what you can see is that the normal mutation burden does show some sort of a trend with age, as we would expect. There is kind of variation from biopsy to biopsy in the mutation burden within the normals, but this is considerably increased in patients with chronic liver disease. And in fact, we find an increase in the mutation burden in patients with chronic liver disease of around about 1,200 mutations. So by the time that you're kind of 60, 70, your liver cells will have, somewhere between 1,500 and 2,000 mutations on average. But we're adding an extra 1,200 in patients with chronic liver disease. And this varies quite extensively from patient to patient, as you can see. And it's not just increase in base substitutions, we also see an increase in indels, and most strikingly, we see an increase in structural variants. And one of the real surprises to us when we looked at this liver data was just how frequent chromothripsis was in these patients with chronic liver disease, considerably more frequent in chronic liver disease than in healthy livers. But we see, kind of complex structural variants like these here with kind of copy number changes associated that is typical of chromothripsis. So we know that chromothripsis contributes to about 50 to 80% of hepatocellular carcinomas. And what's really striking is that it's already evident in chronic liver disease, particularly alcohol-related liver disease. So when we look at the clonal structure, what we see in healthy liver is, as you would imagine, sort of phylogenetic trees that look like this. Essentially, they look like cones with long, parallel, and independent branches. And when we can map each of these clones onto the liver biopsy, because we know where each of the biopsies, the microdissections that we've sequenced came from in that liver biopsy. And you can see that they're essentially each of these regions that we've microdissected is independent of every other region, with the exception of this piece, these clones here, which are clonally related. But by and large, each of these regions is clonally independent of one another. So healthy liver, we see this repeatedly across the healthy livers. So healthy liver has this tightly knit polyclonal structure. Each clone comprises only a few hundred to 1,000 cells or so, which suggests that each liver cell is probably only undergoing a handful of cell divisions throughout adulthood, really fairly quiescent tissue in the healthy state. In contrast, when we look at liver cirrhotic tissue, we find a completely different structure. So we see in the phylogenetic tree much more structure with more clonal relationships between biopsies that we've sequenced, microdissections that we've sequenced. And then when we layer these clones onto the histology, you can see that these clones span quite large distances. I mean, here's a massive clone in this liver, another one here that's taking up this entire cirrhotic nodule. All of the cells in this cirrhotic nodule are essentially derived from the same ancestral cell. But what's interesting is that these clones respect the nodular architecture. So you can see this fibrosis surrounding the hepatic nodules, and essentially this fibrosis is walling off each of these clones in the liver disease. So each of these essentially becomes its own little empire that is where there's competition, clonal competition within the nodule, but no competition between nodules because the clones can't cross the fibrotic bands, as it were. So really quite a rich picture, and certainly very different clonal structure to what we see in healthy liver. So, I mean, this is a conference about mutational signatures, so it would be unwise not to talk about mutational signatures in this data. And of course, it turns out to be quite a rich landscape of mutational signatures that we see in liver disease. Essentially what we see is that in healthy liver, most of the kind of clock-like mutational processes that are seen in all tissues and all cancers and accumulate at that linear rate with age, those dominate the picture in healthy normal liver, although there are still occasional patients, even with normal liver, who have signatures, other unexpected signatures. And this bright yellow signature is the signature of aflatoxin. And it matches perfectly the aflatoxin signature. And this one healthy patient, one has a few regions of his liver that show the signature of aflatoxin exposure. Interestingly, aflatoxin was first discovered as a contaminant of moldy grain in farmers in East Anglia in the UK, and in arable farmers in East Anglia. And it turns out that this patient is in fact an arable farmer in East Anglia of the UK. So it's possible that actually this is an occupational exposure to aflatoxin from the Aspergillus spores contaminating the grain. In the setting of chronic liver disease, as you might expect, because we see increased mutation burden, what we see is that much of that increased mutation burden is coming from additional mutational processes that we don't see as much of in the healthy liver. So I'm going to pull out a few of these in more detail. So one of the signatures that we see is signature 4, using the COSMIC numbering system, and signature 4 is typically seen in lung cancers, and particularly in tobacco smokers, and it is seen in liver cancers, although its relationship to cigarette smoking is less well defined in that setting. And it's really interesting, we see it in quite a few patients with chronic liver disease, and particularly striking in this patient, who it was indeed a heavy smoker and a heavy drinker. And what I'm showing here is a phylogenetic tree like I was showing for the clonal structure of the cirrhotic liver, but now I've layered onto this phylogenetic tree the proportion of mutations on each branch that can be attributed to the different signatures that are active. So a couple of the sort of background liver signatures that we see in all of the samples, but this sort of, I don't even know what color that is, sort of plum-colored band stripe represents the mutations that we're attributing to that tobacco signature. And what's really interesting here is that this patient there are a number of the microdissections, number of clones here that will have 5 to 12,000 mutations, and then about half the clones have a relatively normal mutation burden. And what you can see is that all of the clones that have elevated mutation burden essentially have a few thousand mutations that are attributable to this tobacco signature, but these ones that have relatively normal mutation burden have much less contribution from the tobacco signature. And when we layer these microdissections onto the histology of the image that we've cut, essentially the section that we've cut, and we then kind of color them. So blue means there's coming from these clusters that don't have much in the way of tobacco signature, and then red means more and more tobacco signature. What you can see is that the clones that have very high mutation burden from this signature are living right next door to clones that essentially have not been touched by this mutational signature. And we really don't understand why you would have two such closely many clones living right next door to one another, essentially in the same person's liver, exposed to the same chemicals and everything else that that person is indulging in. And yet some of them get several thousand mutations from the exposure, and then others are basically untouched by it. We have no reason, no explanation for this variation. And it's the same pattern. We see one patient who had aristolochic acid signature, aristolochic acid being an important carcinogen in East Asia and the Balkan states, and is known to cause hepatocellular carcinoma. And here again, we see that there's this variation from clone to clone in the number of mutations, the fraction of mutations that can be attributed to this aristolochic acid exposure. And we are at a loss to explain why this might be. We found one novel mutational signature in this liver data that hadn't been described before. This is the signature that's characterized by T2A mutations, particularly in a CTG context, with quite strong strand bias, as you can see, suggesting that adenines are the targets for the mutation. And what's really interesting about this signature is that it sort of resembles some of these signatures that Serena Nixanel and Dave Phillips pulled out in their in vitro exposure study that have this kind of similar pattern, but we don't really know whether this is a coincidental similarity or somehow these toxins may be contributing to this signature that we see in some of the patients with chronic liver disease. What's fascinating about this signature when we look is that again, this is the same pattern. We're seeing it again in some regions of the liver, but completely absent in other regions of the liver. These clones are really very variable in the frequencies. You can see here's the sort of mutational spectrum of a microdissection that has very low levels of this. This looks like any normal liver mutational spectrum, and here's a neighboring microdissection with very high levels of the signature. I mean, it's just, you can spot it by eye. What's really interesting is that when we layer this signature on the phylogenetic tree, as is done over here, what you can see is that the purple signature, which is this novel signature, is really profoundly late. So we see it, if you look at this particular clone here, the parental clone barely has any of this mutational signature, but the two daughter clones, subclones of this original parental clone, have a large majority of the mutations coming from this novel mutational signature. So we think it's a relatively late onset mutational signature, possibly as the patient is becoming established with chronic liver disease and then the liver's losing its ability to metabolize xenobiotics, and perhaps that's why suddenly these exposures might become more pronounced in terms of their mutational signatures. So I think when we think about mutational signatures, which is of course the focus of this meeting, it's also worth kind of also pondering what these mutations are doing to the cells. And so, you know, when we think about cancer evolution, you know, we have to get the mutations causing a sort of background genetic variation within the samples. And then those mutations, essentially, some of them have to change the biology of those cells, and those we kind of understand as driver mutations. So we have done a driver mutation analysis for the 1,500 genomes that we've done. And what's really intriguing is that 3 completely novel genes popped out of this analysis, one of which was FOXO1. Now, FOXO1 is the major transcription factor downstream of insulin signaling. So insulin binds its receptor, and then you get AKT activation, and AKT phosphorylates FOXO1. And then these 14-3-3 proteins export FOXO1 from the nucleus by binding to the phosphorylated FOXO1. And the place that they bind is this particular motif, and we write next door, so that it's this threonine 24, which is phosphorylated by AKT, and then for the 14-3 proteins bind this 6 amino acids here. And the mutations we find are all at this serine 22, and they change it from a serine to a bulky tryptophan. And we see this exact same hotspot in multiple patients within our cohort. And the prediction would be that essentially the mutations block the export of FOXO1 from the nucleus. So insulin would essentially no longer work to export FOXO1. And indeed, that's what we see when we do in vitro studies. So if you look here, in the wild type FOXO1, when you give it insulin, the GFP nicely leaves the nucleus. As you can see here, the blue becomes much more apparent. But when we look at the mutant FOXO1, it stays well and truly stuck in the nucleus. It can't be exported. So essentially the prediction is that these mutations render those hepatocytes completely resistant to, well, not completely resistant, but very resistant to insulin activity. And indeed, we see transcriptional consequences of insulin resistance and metabolic programs when we look at metabolites in cells that have these mutations. We see essentially the signatures of insulin resistance in these hepatocytes that have these mutations. What's really striking is that these mutations are being seen multiple times independently in the same person's liver. So here's someone who has four separate independent acquisitions of the exact same mutation and an adjacent hotspot mutation. Here's another patient who has 3 independent clones with exactly the same FOXO1 mutation. And indeed, of the 7 patients who had these FOXO1 S22W mutations, 6 of those patients had the mutations in 2 or more independent clones, and 1 of those patients had 9 independent clones with the exact same mutation. So this is convergent evolution at a massive scale. Really interestingly, we do not see these mutations in either normal liver or in hepatocellular carcinoma. So the selective advantage that they're conferring is really very specific to the phase of chronic liver disease that we're studying here. And the pattern's repeated in the other 2 genes that we found. So SIDE-B is a master regulator of lipid droplet fusion in hepatocytes, and the mutations we see cluster in these domains and often change the charge of the amino acid, and it's predicted essentially to block the function of SIDE-B. And what SIDE-B does is brings together lipid droplets for fusion, and indeed when we study the effects of these mutations, we block this lipid droplet fusion in the hepatocytes. And we see again the same pattern of multiple independent clones within the same liver biopsy showing these mutations in this gene, another instance of convergent evolution. And again, one patient had up to 14 different clones with SIDE-B mutations. Again, these mutations are not seen in healthy liver, and they're not seen in hepatocellular carcinomas. And interestingly, the knockout mouse for this gene is resistant to fatty liver disease, essentially. And the third gene that we found is GPAM, which is the master regulator in converting free fatty acids to storage triglycerides in the liver. And again, we see clustering of mutations in the relevant domain of this protein, or we see nonsense frameshift mutations, so predicted loss of function mutations. Again, lots of evidence for convergent evolution. Here's one patient who has no fewer than 7 different GPAM mutations within his liver, all acquired independently of one another. And like we see for the other genes, these mutations essentially are protective in the knockout mouse model. They're protective against fatty liver disease and steatohepatitis. And again, these mutations are not seen in healthy liver, and they're not seen in hepatocellular carcinoma. So really quite a striking coalescence of these features. So within a patient, the same gene is often hit many times, but between patients, we tend to see often different types of genes being mutated. So what's going on? So essentially what we think is going on is that in the healthy context, you have this kind of ongoing evolution of genetic diversity. All of the mutational processes and signatures that we've discussed are kind of leading to cells with many different genotypes. And then essentially along comes a disease, some sort of selective pressure or toxicity, and that kills a whole bunch of normal or wild type cells. And only those cells that have some particular genetic change that allows them to escape that toxicity can survive that selective pressure. And it may well be that the selective pressure is different from patient to patient. So, in one patient, you get pink genes being selected for. In the other patient, you get blue genes being selected for. And then over time, only those surviving clones can expand. And so you wind up getting these large clones with this pattern of convergent evolution of the similar sort of patterns of driver mutations like those that we see. And it may well be that these mutations just don't progress to give liver cancer, particularly in this setting, because these genes that confer a selective advantage in the setting of chronic liver disease, which is often a setting of caloric excess, are all pointing at fat metabolism being a critical pathway of toxicity in these diseases. But that pathway may not confer a selective advantage on clones, allowing them to become malignant. And so these mutations may actually protect against liver cancer while potentially and protecting the hepatocytes against the kind of disease toxicity. What we have no idea about is whether these drivers actually affect the metabolism of the liver as a whole. You know, if you have hundreds of grams of liver tissue that have these mutations through convergent evolution, does that lead to organ dysfunction? We just have no idea at this stage. So the final piece of data that I wanted to mention is that what we find when we compare hepatocellular carcinoma from the same individual to their background liver is that we find that typically hepatocellular carcinomas have an elevated mutation burden compared to the background liver. So there seem to be even more mutations in the cancers. So that's really where I want to finish, and particularly to thank Simon Brunner and Stanley Ning, who drove the liver work with help from FOAD and Natalia, and my long-term collaborator on the liver, Matthew Hort. Thank you very much.
Thank you, Peter, very much. I see that we have a few minutes, and maybe we could ask you a couple of questions from the Q&A. So let me start from this one. Is excess NAT consumption linked to the mutational signature of aflatoxin you observe in AFLD patients?
So the signature of aflatoxin exposure, we only saw it in one individual, in that one healthy individual. He wasn't a particularly heavy drinker. We did not, when we looked at, so we had in the cohort, we had a mix of patients with alcohol-related liver disease and non-alcoholic fatty liver disease. When we looked at the signatures between the two groups of patients, we didn't see any differences. So there has been some possibility that, signature 16 is related to alcohol. And there was there was a sense that it might be a little bit more enriched, but it wasn't statistically significant. I think we don't have quite enough patients at this stage to really answer that question definitively. But I think it remains interesting. One of the problems that we have is that we're relying on kind of self-reported alcohol intakes in these individuals, and I'm not sure that self-reporting is always very accurate. And of course, many, particularly in the setting of chronic liver disease, many people with non-alcoholic fatty liver disease will still have some alcohol intake, and similarly, people who have alcohol-related liver disease often have obesity and metabolic syndrome that goes with it. So separating the two disease entities can be difficult. I think we might see a more striking difference in mutational signatures if, for example, we had sequenced patients with chronic liver disease arising from, let's say, chronic viral hepatitis or autoimmune hepatitis, and I mean, we'd like to do those studies. We're just kind of launching those at the moment, but I think watch the space essentially.
So let me ask another question. Isn't the whole genome sequencing at such low depth, 15x, resulting in a lot of noise in the final code variants?
So we sequenced, so the sequencing depth that we aim for is 15x when we do single cell derived colonies, which we know are completely clonal because they're derived from a single cell. In the liver samples, we aimed more for 30x coverage. Essentially our accuracy comes down to our accuracy for the mutation calling. So we have various ways of measuring the accuracy of the variant calling. One way is that we can take two adjacent sections of the liver and essentially cut the same microdissection from each section and then sequence those independently. And because the clones are sort of big enough in chronic liver disease, we can look at, how often do we call the mutations in each one. And what we estimate is from when we do that, so they're essentially independent replicate experiments, we estimate that the sensitivity is probably around about 85–90% at 30x sequence depth, and the specificity is about 95%. Obviously, there's a trade-off between sensitivity and specificity, but it works out to be surprisingly good. It's all about getting the microdissection small enough that you're sequencing something that's relatively clonal.
It's very interesting that the clones respect the nodular boundaries in cirrhotic livers. Do you think that similar histological features, such as fibrosis, shape spatial evolution in other cancers by limiting clonal mobility?
Yes, I do think that, and I think that is. I think if one thinks about the tumors which have a very florid stromal reaction, so there are subtypes of mesothelioma that have a very florid stromal reaction. Pancreatic carcinoma is notorious for having lots of fibrosis and fibroblast proliferation. I think that those stromal reactions, there's been a lot of debate about whether they're tumor suppressive or tumor promoting. But I definitely think that it's quite likely that those kind of stromal reactions do shape the way that the cancer cells invade through tissues when they get that potential. Of course, here by definition, these nodules are not invading through tissues, and the clones. The other thing that's interesting about the fact that the clones respect the nodular architecture is that it suggests that the fibrosis is being laid down quite early in the disease process. If it was a very late event, then one might expect to see rather more sharing between the nodules. But my guess is that this fibrotic tissue is already restricting clonal expansions at relatively early stages of disease.
We have time for just another one, I guess. In your approach, how reliably can be tobacco smoking signature in the liver discerned from other G2T inducing processes, such as oxidative DNA damage and the resulting the SBS18, the reactive oxygen species?
Yes. So I think that's a really interesting question. And I called it the tobacco signature. In fact, I should have called it signature 4, and because it is a leap to say that it's tobacco. In fact, it may, what I can say about it is that the in vitro studies essentially say that most of the signature is a polycyclic aromatic hydrocarbon signature. So the polycyclic aromatic hydrocarbons bind to the guanines and lead to mutations, and that pattern of mutations is definitely distinguishable from reactive oxygen species signatures using the sort of experimental evidence that I cited earlier in the talk. So I'm reasonably confident that what we're seeing is not reactive oxygen species. It's quite likely that this is something related to polycyclic aromatic hydrocarbons. What I will say, though, is that there are many sources of polycyclic aromatic hydrocarbons that are not tobacco related. So obviously, air pollution is a source of polycyclic aromatic hydrocarbons. Diet can be as well. So burnt meats have polycyclic aromatic hydrocarbons, and of course, both air pollution. There can be some quite large particles that will deposit on the upper GI aerodigestive tract and then get swallowed. And similarly for dietary sources of polycyclic aromatic hydrocarbons, those too will be swallowed. And then what will happen is that they will get absorbed through the gut, and their first place of landing after absorption will be the liver through the portal circulation. So it's entirely feasible that at least some of the signature that we're seeing, that I loosely called a tobacco signature and should have called signature 4, is actually a signature of polycyclic aromatic hydrocarbon exposure that's coming potentially from sources other than tobacco smoke. Although, it may well also, in that particular individual that I showed, who was a heavy smoker, could come from swallowed or inhaled and absorbed tobacco smoke carcinogens.
Okay, thank you. Thank you very much, Peter.
I will stop sharing and hand it back to you.
We'll move now to the first section.